Okay, just a quickie here. This one is a bit of terminology that we use to describe DNA. DNA is described as universal, non-overlapping, and degenerate. Okay, so we're going to look at these terms one at a time and see what they mean. So we've got universal. Well, universal basically means that all the living things that we know about use DNA. Some viruses, it said living, viruses not considered to be living, use RNA as their main genome, but they still convert into DNA in the host organism. So DNA is universal. But what we really mean by universal is that each triplet code or codon codes for the same amino acid in all living organisms. So if GAA or GGT means glycine in one living organism, it means glycine in another living organism. Okay then, so non-overlapping. This basically means that each triplet and each gene is separate from each other. You don't get any overlap between one triplet and the next triplet. So if we draw a stretched out piece of DNA, obviously it's all coiled up around histones in chromosomes, in the nucleus, we know that. But if we were to pull it out as a single string and we were to look at, we'd probably have a gene here, but we don't have another gene starting here and finishing here. That's not possible. It's non-overlapping. You might have some of the repetitive non-coding regions in the middle. You might have a long gap. This might be the next gene, might be a smaller gene. You might have then obviously going to have another gap, and we might have a longer gene here. But this is going to be a gene, this is going to be a gene, this is going to be a gene, but they don't overlap. The same would go if this was on a smaller scale with DNA triplets. And degenerate, the one this is probably the least obvious. Degenerate means that there are more DNA triplet codes than there are amino acids. And so the same amino acid can be coded for by more than one triplet. So it's possible that if GTT codes for glycine, maybe GCT also codes for glycine. It's not one-to-one, triplet to amino acid. Whilst we're doing some definitions, there's a couple of others that we're going to be super useful whilst we're doing this. We're going to do the genome. Much more commonly in the news these days, the genome rather than genes. So the genome is the complete set of genes in a cell or an organism. All of them. And something you might not have heard about is the proteome. Well, the proteome is the full range of proteins that a cell can make. Obviously, the gene holds the code for making the proteins, and we're going to look at that next in protein synthesis. And the proteome is the complete range, the full range of proteins that a cell can make. So these are all going to be needed during the course of understanding these at the very least. Using them maybe a little bit less. I'll just give you a quick overview of protein synthesis. There's two stages of protein synthesis, and we're going to look at these next, but this will give you a useful heads up. So there's two major stages. You've got transcription and translation. Now, these are very commonly confused. Students get them mixed up all the time. I suggest you spend 30 seconds making an association to get this into your head. But transcription is like a scribe. So we're going from DNA into RNA or messenger RNA. And this DNA is made of nucleotides, and messenger RNA is made of nucleotides, so this is like a scribe. The language isn't changing, it's staying in the same form. Let's do DNA can be blue, and then it changes into red, but it's still nucleotides. Now, translation, we're taking that messenger RNA, moves into the cytoplasm, we're going to go into the details later, and it's converted into amino acids or polypeptides or proteins. These are all kind of interchangeable, really. I'm going to put all three. So here we're changing the language. We're going from our nucleotides, and we're converting it into a chain of amino acids. This is obviously going to be really long. The chain of amino acids is then going to fold up, and we're going to have maybe our alpha helices, we've got maybe some beta pleated sheets, and this is going to fold up into our protein. And obviously, we need to maybe do some joining up of the dots, but I'll leave it so you can see the regions. Okay, so this is translating. We are literally, it's a translation from one language into another language, because we're going from nucleotides into amino acids. Here I'm going to put alpha alpha because I'm lazy like that. In my notes, I use alpha alpha for amino acid all of the time. But yeah, we're changing the molecules which we're made from. So I hope that helps. We're going to look at protein synthesis next. So for the full detail, check that one out.